Hi guys, and a very good day to the Miss Era and everyone's here. So today, we from Group 5 will present to you the topic about muscle contraction, which is under the topic nervous and muscular system. You guys can refer this topic in chapter 39, page 721 until 723 in media textbooks. First of all, let me introduce to you our presenter today. So here we have Ismi and Shira, which is the one who is talking right now, Daniel Haqeem, Fadz, and Hawa. Next, learning outcomes for this topic are be able to identify what is the component that involved in muscle contraction. Second one, describe the role of calcium and myosin in muscle contraction. Explain the use of ATP in muscle contraction. And the last one, describe how thick filaments be functioning in muscle contraction. Alright, let's move on to the next slide. So here we have the introduction of muscle contraction. As we know, muscle is a part of our body that helps to move blood throughout all of our body. But today, we want to go more detail on muscle contraction. Muscle contraction is the tightening, shortening, or lengthening of muscle when we do some activity. For example, when we hold or pick up something, muscle contraction happens on our hand. Next, let's take a look at the component of muscle contraction. Here we have 4 components, which are actin filament, calcium Ca2+, myosin filaments, and ATP. So, it is good for us to find out what is the function of each component, right? Well, as we can see here, actin filaments will slide past myosin and causing contraction. Ca2+ is needed for myosin to bind to actin filaments. Myosin filaments will pull actin filaments by means of cross bridge, which are enzymatic and split ATP. And the last one, the function of ATP for sure to supply energy for muscle contraction. From the picture here, we can see the cross bridge when myosin filaments pull actin filaments. And we can see that ATP split out to produce ADP. Next, I will pass to Hawa for further explanation.
Hello, my name is Hawa binti Azan and my metric number is AS12696. I will explain about the uses of ATP in muscle contraction. ATP provides the energy for muscle contraction. Although muscle cells contain myoglobin, a molecule that stores oxygen, cellular respiration does not immediately supply all the ATP that is needed. In the meantime, muscle fibers rely on creatine phosphate or phosphate creatine, which is a storage form of high-energy phosphate. Creatine phosphate cannot directly participate in muscle contraction. Instead, it anaerobically regenerates ATP by the reaction where phosphocreatine reacts with ADP and produce ATP along with creatine. This reaction occurs in the midst of sliding filaments, and therefore, this method of supplying ATP is the speediest energy source available to muscles. When all of the creatine phosphate is depleted, mitochondria may by then be producing enough ATP for muscle contraction to continue. If not, fermentation is the second way for muscles to supply ATP without consuming oxygen. Let's continue for fermentation. Fermentation, which is occur when strenuous exercise first begins, supplies ATP for only a short time and lactate builds up. We all have had the experience of needing to continue deep breathing following strenuous exercise. So this continued intake of oxygen, which is required to complete the metabolism of lactate and restore cells to their original energy state, known as oxygen debt. Next, the lactate is transported to the liver, where 20% of it is completely broken down to carbon dioxide and water. The ATP gained by this respiration is then used to reconvert 80% of the lactate to glucose. In persons who regularly exercise, such as athletes in training, the number of mitochondria increases, and muscles rely on them rather than on fermentation to produce ATP. Hence, less lactate is produced and there is less oxygen debt. That is all for the uses of ATP in muscle contraction.
Assalamualaikum, my name is Muhammad Fadz and I will continue the explanations about muscle contractions with role of calcium and myosin in muscle contractions. So, functions of calcium. The placements of the other protein associated with thin filament. This structure composed of a double row of twisted actin molecules. So at the first structure, tropomyosins, actin filament, and also troponin. So with the calciums, when released, calcium will bind to troponin. This exposes myosin binding sites. Next. So next, we have the functions of myosin in muscle contractions. So the first picture, ATP is harassed when myosin head is unattached. The second picture, myosin heads bind to an actin filament. Later, a power stroke causes the actin filament to move. Last but not least, when another ATP binds to myosin, the head detach from actin. So the cycle begins again. Although only one myosin head is fractured, many heads are active at the same time.
So next, I will explain about the T filaments. T filaments is a bundle of myosin molecules with double globular heads. As you can see in this picture, they have the upper layer and lower layer of thin filament. Myosin heads function as ATPase enzymes, splitting ATP into ADP and phosphate. This part we will discuss in the next slide. So the next slide. So this part we will learn about the cycle of the filament movement approach the center of the sarcoma. Firstly, the reaction of the enzyme ATPase. Enzyme ATPase is the function is to splitting the ATP into ADP and phosphate. The enzyme ATPase will activate the myosin head to bind actin. Initially, ADP and phosphate remain at the myosin head until the myosin head attached to the actin, and it will form a cross bridge. Now ADP and phosphate are released. This causes the cross bridge to change their position. The thin filament will be attracted towards the center of the sarcoma. When more ATP molecules are bound to the myosin head, the cross bridge is served when the head is released from the actin. So the cycle is repeated. Each time the active filaments move near the center of the sarcoma. So to make guys more understand about this part, I will share to you this video from the Connect explain about this part.
Each skeletal muscle of your body contains muscle cells called muscle fibers. Within each muscle fiber are many myofibrils divided into sarcomeres. Sarcomeres are the contractile unit of a muscle. When the muscle contracts, the sarcomeres shorten by sliding the actin filaments past the myosin filaments. Let's take a closer look at how this happens at the molecular level. Here we are taking a closer look at the actin filament, sometimes called the thin filament. As we can see, two other proteins are associated with the actin filaments. Threads of tropomyosin wind around the actin filament, and troponin occurs at intervals along the threads. When the nervous system signals that the muscle needs to contract, calcium ions, shown as Ca2+ in this diagram, are released from the sarcoplasmic reticulum. The ions combine with troponin, which then shifts the position of the tropomyosin threads. When tropomyosin moves out of the way, myosin binding sites are exposed along the actin filament. The heads of a myosin filament, the part that looks like a golf club, have ATP binding sites. When ATP binds to these heads, an ATPase enzyme splits ATP into ADP and phosphate. Remember that the calcium ions expose the myosin binding sites on the actin filament. The breakdown of the ATP on the myosin filament allows the myosin heads to attach to an actin filament. This forms a cross bridge between the two filaments. Once this cross bridge is formed, the ADP and phosphate are released, and the cross bridge changes its position. This is the power stroke that pulls the actin filament toward the center of the sarcomere. When ATP molecules bind to the myosin heads, the cross bridges are broken, and the heads detach from the actin filament. This completes one cycle of the contraction. If calcium ions are still present, the cycle repeats itself. Each time the cycle is repeated, the actin filaments move closer to the center of the sarcomere. When the nervous system wants to stop muscle contraction, the nerve impulses cease. The calcium ions are then actively transported back into the sarcoplasmic reticulum. The contraction stops, and the muscle relaxes.
So next part. Okay, the next part, there are two factors that make the muscle contraction stop to undergo. So the first one is when the nerve impulse is ceased, and also when the calcium ions are returned to their storage site. Calcium ions can be pumped back to the calcium storage site by using active transport proteins found in the sarcoplasmic reticulum membrane. So the last part, what happens if a person or an animal dies? So the first one, ATP production will cease, so that without ATP, the myosin heads cannot detach from actin, nor can calcium be pumped back into the sarcoplasmic reticulum. And thus the muscles remain contracted. This phenomenon we call as rigor mortis.
In summary, muscle contraction is really important, especially for an athlete. Why? It is because of the movement. In addition to movement, muscle contractions also fulfill some other important functions in the body, such as posture, jaw stability, and also heat productions. In this presentation, we can see that the functions of ATP and also role of calcium and myosin in muscle contraction. In these presentations, we refer to media and also these links about to enhance our knowledge in muscle contractions. So, credit to Ismi Shira binti Muhammad Jamri, Hawa Adani binti Azlan, Muhammad Fazil bin Ahmad, and also Daniel Haqeem bin Muhammad Rizal that successfully done these presentations. That's all. Thank you.